Okay, Tim, we're good to go.
Hi. Oh, hi. Hi. Welcome to today's open research webinar by eLife. We're aiming to give early career researchers an online platform to continue to share their research during the COVID era when people can't go to meetings. So I'm Tim Behrens, and I'm deputy editor at eLife. And I'm a neuroscientist. And so today we're hearing from Uri Maoz from Chapman Uni in the US, from Stefania Mattioni from UCLouvain in Belgium, and from Anantha Sundararaman from Bristol in the UK. After each 10-minute talk, we're going to have 5 minutes questions for the speaker, and then we'll move on to the next talk. So to ask a question, you can type it into the Zoom chat directly, or you can put it into the Google document that's linked here. So the link is also in the chat. And so we're joined today by Anya and Naomi from eLife, who are working in the background to support. And so they're going to help line up your questions. So if you're able to, I'll invite you by name to ask your question, and the team will unmute you so you can do that. Otherwise, I can read your questions out loud and include your name where possible. The open document is also a place for you to contribute shared public notes. So we welcome that, and to list yourself as a contributor in the list above the speakers for today's webinar. So thanks very much. Finally, I'd just like to let you know that we're recording the webinar and also live streaming it to YouTube. During this webinar, we ask you that you please be respectful, honest, inclusive, accommodating, appreciative, and open to learning from everybody else. Don't attack, demean, disrupt, harass, or threaten others, or encourage such behavior. If you feel uncomfortable or unwelcome on any of these webinars, please contact eLife by email to events@eLifeSciences. This inbox is being watched by Miranda and I at eLife. The eLife organizers reserve the right to ask anyone to leave or deny access to any subsequent webinar or Zoom request if they're being unhelpful. So if you need any help at this point, please send Miranda, Naomi, or Anya a chat message directly using Zoom. Okay, great. First, we're going to hear about Uri, and he's going to take over the screen. He's talking about neuroscience of volition for decisions that matter. Thanks very much indeed.
Thank you very much, Tim. So yes, my name is Uri Maoz from Chapman University, as you were told, and I'll be talking about the neuroscience of volition for decisions that matter. Now, if I asked any one of you to raise one of your hands, you, of course, would be able to do so. And the order of events, as you probably experience it, is that you consciously decide which hand to raise, and then that causes you to raise your hand. So if you looked at the intuitive order of events, there's the conscious decision, then sometime later there's information in the brain about the action, and then there's the action itself. However, an experiment in the 1980s by Benjamin Libet and colleagues cast some doubt on that order of events. In that experiment, or at least one version of that experiment, Libet asked the participants to raise their hand whenever they had the urge to do so, and at the same time to look at a clock that had a rotating dot, and then just say where the dot was when they first had the urge to move or when they decided to move. So the dot started rotating, and then at some point they had the urge to move, and maybe they said that the dot was at 5 when they had the urge to move. Now, at the same time, EEG was recorded from the participant's scalp, looking at electric activity in the brain. And something called the readiness potential was calculated from that EEG. And that readiness potential is typically associated with voluntary movement. So this is here the readiness potential. This is where movement occurs. And maybe somewhere around here, something like 1 second before the movement, we would say from the readiness potential that we see something. There's some information in the brain about the upcoming action. However, where did people report that they had the urge to move or decided was around here, so about 200 milliseconds before movement onset. So the strange thing is what is going on in this time range here, because on the one hand, we already have, as we said before, information in the brain about the action, but the subject has not yet reported that they have decided to move. So is it the case that the decision was made unconsciously? So these limit results caused quite a stir. They have been cited a few thousand times already, and they were mentioned widely in the lay press. And I should also note that they were not a fluke. This holds for if you ask people to raise one of their hands, or if you ask them to raise either one of their hands, so when to move or which hand to move, these precursors of movement were found actually much earlier using fMRI, even a few seconds before movement onset. They were found in single neuron recordings in humans as well. And we found some neural precursors of action even online in real time, so on the fly as things happen. So some people have concluded from those Libet results that the decisions in Libet experiments were made unconsciously. From that, they concluded that perhaps generally decisions are made unconsciously. And from that, it was suggested that, maybe this is a challenge for some of the social pillars, like our notion of free will and maybe even moral responsibility. If we are doing everything unconsciously, where is our free will and how are we morally responsible? So the thing I wanted to tell you about today is something that we've looked into regarding this arrow here. The thing that troubled us was that the Libet experiments, as I told you about them, were about raising your right hand or your left hand, or or flicking your wrist or something like that whenever you had the urge to do so. But we were wondering whether these results generalize to deliberate decisions. So deliberate decisions could be things that you deliberate about, which route to take to work in the morning, or more important life decisions, which job to take, life partner to choose, and so on. And we were wondering whether these arbitrary decisions and these more deliberate decisions are really the same. And this is work that I've done with collaborator Liad Mudrik and other collaborators. And what we've asked people to do is make donations to different nonprofit organizations, NPOs, and those decisions that they made about these donations actually determined a $1,000 donation. So this was, this was real. It was not fake. So on a trial, subjects might see two nonprofits, maybe the Hunger Project and Cancer Research Institute. In some of those trials, those decisions were hard, and other trials, those decisions were easy for those subjects. And they had to decide which one to donate $1,000 to, and these were the deliberate decisions. Subjects also made arbitrary decisions. Interestingly and importantly for us, the visual input for the subjects were the same, as was the motor output. However, the meaning of the decision was quite different, because this time we told them, no matter what you choose, both NPOs will get $500. So your choice doesn't matter. And so then whatever button you press is completely arbitrary. So if we look at the neural recordings from that, we can see, if you look at the arbitrary decisions, we can see a downward trend that's reminiscent of the readiness potential. But we cannot see the same for deliberate decisions. We see it for arbitrary, we do not see it for deliberate. And the statistics seems to confirm what you see here. And if you look on average over all the subjects, you can see a nice readiness potential for arbitrary decisions, but just a slow downward trend that's not a readiness potential for deliberate decisions. And again, the statistics confirms this, what you see. We ran various control analyses to make sure that these results are not the outcome of some trivial tweak or something like that, and they were not. And interestingly, we also looked at one more thing called the lateralized readiness potential. That's a potential that's more related to motor events. And there you see that arbitrary and deliberate decisions, there is no difference. So this really chimes in with the idea that the motor output here is the same between arbitrary and deliberate decisions. Further on, we worked with some modeling because recently there was a claim that this readiness potential is maybe actually artifactual. It might be due to autocorrelation in the EEG and with the specific method by which the readiness potential is computed from the EEG, rather than actually really being something that tells you that a voluntary action is about to occur. So we took that model that was done by Aaron Sugar and others in 2012, and we extended it to our case, and it fit our data quite well, and from that that model really predicted this slow downward trend in deliberate decisions, but to really see a readiness potential in arbitrary decisions. So to conclude from this, we would say that the neural signals that are associated with arbitrary decisions do not generalize to deliberate decisions, at least in our experiment. And if you think that the limit results pose a threat to our notion of volition, this threat doesn't seem, at least again from our results, to generalize to deliberate decisions. Now, I would arguably say that we care more about these deliberate decisions, the person you're going to live with, things that you think about, especially for a long time, than which hand you're going to raise. Certainly, if you want to talk about moral responsibility, nobody's going to take you to court for raising your right hand and not your left for no reason and for no purpose. So I think our results suggest that the onus is now on those who think that the limit results generalize to deliberate decisions, to show that they do, because our results suggest that they do not. And this is just the paper that that was discussed here, if you want to look more into this. Of course, this was just a brief overview. Now, if I've piqued your interest into the neuroscience of volition, let me just say one more thing, that there is now a large international project that is happening, looking into how the brain enables conscious causal control of action. We've got people from all over the world working together, neuroscientists and philosophers. You can see more information here. And if I may, I'll just say that, like everybody, we are seeking postdocs, PhD students, and RAs, and you could contact me in either one of these addresses for more information. Thank you very much.
Hey, thanks very much indeed, Uri. Very interesting indeed. Gets right to the heart of who we are and what we are. As I say, if you've got questions, you can post them in the chat, or you can post them on the document. And so I guess I'll start with asking Uri a couple while we're while if there are any from the audience. Firstly, did they choose the same things for the two types of decisions? It seems like they might have done to me, or yeah, well,
the same. You mean the same nonprofits?
Yeah, exactly. So you can imagine non-deliberate decisions that are nevertheless favorable because you have simple Pavlovian effects towards things that you like, etc., etc. So I'm interested to know whether those are what were driving the decisions.
Right. So I mean, I didn't get into it, but we can actually, if you look at the consistency, and I guess this is what you're asking about, right? The consistency of the decision. So you can see that there is nice consistency for deliberate decisions, but there was, so I'll explain what I mean by consistency. I didn't go into it in the experiment, but there were two parts of the experiment. In the first part, subjects just rated on a scale of 1 to 7, each one of the nonprofits, how much they would want to donate $1,000 to it. And the consistency here is if something that was, let's say, a 3 came up with something that was a 7, you'd expect them to choose the 7 over the 3. So how consistent were they? So you can see that for deliberate decisions, the subjects were quite consistent. For arbitrary decisions, they were very close to chance. However, if you look at this cross here, they were slightly off chance. So they were just a little bit. Yeah. So if something came up that you really don't like versus something you really like, even though it's arbitrary, it doesn't matter which button you press, it's going to go 50-50 to both of them. You still had a slight preference for the thing that you preferred.
And so, just while we're waiting, it seems to me that is it the case that the deliberate decision is less tied to an action somehow? So, in fact, maybe you've got that here as well. So, is it the decision that is it the case that the deliberate decisions are taking them longer, or somehow there's more delay between the decision and the action on the deliberate decision than there is, or
yes.
So, for example, and so if that's the case, then your ERP can be smoothed over a longer period of time and won't show that. So let's say one trial is really rapid and the next trial takes a lot longer, then once you start averaging the trials together, it's going to be a very different thing from things that are very precisely timed, is it not?
So we've that's one of the controls that we did. We looked into, I mean, one concern that we had ourselves was this due to the reaction time differences? So we did various things to look into that. We took only the longer arbitrary trials and only the shorter deliberate trials, and you still see an effect between them. We also looked at whether there is a trend between the difference in the RP between deliberate and arbitrary, and we could not find an effect. And we did this both within subjects and between subjects, and there were no effects in both situations. So it seems that this does not explain our results. But yes, that was something that came up for us also as one of the first things that we checked, because if then it's less interesting. But again, this was not driving our results.
And yeah, cool, interesting. And so 0 here is the time when they actually make the action, right?
Yes, that's when they press a button. Yes. So
you would, in some senses, you would really expect there to be a buildup of brain activity in the deliberate decision before they make the action. So did you measure the time at which they report to have made the decision? Do you see what I mean?
So that's, I mean, so we didn't, and we didn't do that for two reasons. One, I don't think that right now there is a good measure of when people make up their mind. Libet's clock is a very flawed idea. There are some other things. We're actually trying to develop some of those things in my lab, but that's one reason. The other one was that we really wanted to try and keep this more like the real world. So it wasn't, I mean, asking people, when did you make up your mind, feels very unnatural to us. So we did not ask them when they made up their mind. But in other experiments, when we try to compare between arbitrary and deliberate decisions about when people make up their mind, we found some differences, but they were very small. They could not have, I mean, this was not like the 1 second difference that we see here. This could not have explained these results.
Cool. That's really interesting. So we're going to move on now, but thanks very much for a great talk. We're moving on to Stefania now, who's going to tell us about categorical representation from sound and sight in the ventral occipito-temporal cortex of sighted and blind. Thanks very much.
Thank you. Okay, hope you can see my screen.
Yeah,
okay.
Perfect.
Hello, everybody. My name is Stefania Mattioni, and I'm from the UCLouvain. I am postdoc at the UCLouvain in Belgium. And today I will speak about the categorical representation in the occipital cortex of sighted and blind. So the world around us is full of rich and dynamic visual stimuli. And to avoid an overload of information, our brain tends to group them into categories. The main one are the animate and inanimate, and then also it creates some other clusters within these two. One of the main regions known to be involved in the processing of this visual categorization is the ventral occipito-temporal cortex, which is a region in which we find clusters of voxels that show a selective preference for one of these categories compared to the others. But what drives the categorical organization of VOTC? In other words, what makes a part of this region to prefer stimuli such as faces and another part, for example, to prefer stimuli such as houses? There are actually two main points of view. The first one is that the low and middle level visual properties of the stimuli are actually driving this categorical organization, such as the eccentricity or the rectilinearity properties or the real world size, so being small or big. On the opposite side, we have the idea that some more high-level functional properties of these stimuli, not purely visual, might drive this categorical organization, something like the animacy of the stimuli or their categorical function. It is not easy actually to test this second hypothesis because it is hard to isolate the high-level function from the low and middle-level visual properties of these stimuli, because normally all the stimuli share these low and middle visual properties within each category. We thought that a good way to test this hypothesis could be to present the categories not in the visual, but in the auditory format, and to present them in a group of people that are born blind. Our hypothesis was that if the categorical organization of VOTC is not driven purely by visual properties, but also by high-level functional properties of these stimuli, we should find a similar categorical organization also in the VOTC when the stimuli are presented as sound, and even in people that never used vision functionally. So to test this hypothesis, we included three groups in our study. A group of sighted people that performed the visual version of the experiment, 17 early blind participants that performed the auditory version of the experiment, and then an additional group of sighted that performed also the auditory version of the experiment. And we presented our participants different stimuli from the main animate, inanimate categories, including the humans and animals, manipulable object and big object categories. Each of these four categories was including two subcategories, as you can see in this slide. And we presented the stimuli while the participant was lying in the fMRI, either in the visual or in the auditory format, according to the version of the experiment. And then we looked at the reaction of the brain to these stimuli in the three groups. So with a special focus, of course, in VOTC. So the first question we had was to look at the general topography of this region. Would the topographical map of VOTC be similar across the three groups? To test this, we basically select each voxel, each small part of VOTC, and we assign to it one of these colors according to which categories this voxel was preferring more than the others. So when we look at the map that we get in the sighted for the visual stimuli, we see the classical map, the well-known map, with the medial region in blue, preferring big objects, and then a bit more laterally in pink, the region preferring face, and then in yellow, the region preferring animals, and finally, most laterally in green, the region preferring manipulable objects. What is interesting to see is that we also found the topographical map when the categories were presented as sound in sighted. And even though these clusters are not exactly in the same position as the one that we see in the visual map, we see that there is still a highly significant similarity between these two maps. And this similarity was even more striking in the case of early blindness. So this analysis is actually telling us that there is a shared similarity, at least in the topography of VOTC. But we wanted to go more in deep to understand if, for example, VOTC really contained information about our categories. Would VOTC being able to discriminate each of our subcategories to the others, for example? To address this question, we used a more advanced multivariate fMRI technique, in which we basically compare the brain activity for each of our pair of categories. And we repeated that for each possible pair, that with 8 categories means 28 pairwise decoding. And in here, higher is the gray column, higher is the ability of VOTC in the subject to discriminate the 2 categories. And then we repeated this analysis also in a control region, the early visual cortex, which is a region known to process low-level visual properties, and will be the control region in most of the analysis we did. So first of all, what we did, we averaged these 28 values to look at the general ability of the VOTC in our groups to discriminate these categories. And what we found is that in the visual experiment, as expected, both early visual cortex and VOTC show a high accuracy in the discrimination of the categories. But we were more interested in the auditory experiment, and here are the results. What we found is that, first of all, in the early visual cortex, only the blind can decode significantly the different categories, significantly better than chance and significantly better than the sighted. But the interesting part for us was to look at VOTC. And what we see is that this region can decode the different categories, both in blind and in sighted, significantly better than chance. Even though we see that the accuracy in the blind is significantly higher compared to the sighted, suggesting that there is an extension of this ability in the case of early blindness. So this analysis is actually telling us that yes, VOTC contains information about different categories, not only when they are presented visually in sighted, but also when they are presented in the auditory format, both in blind and in sighted. Does this mean that the functional profile of this region is similar across the groups? And first of all, what do I mean with functional profile? If we go back to the 28 pairwise decoding, what we see in each group is that there are some categories that are more easily decoded compared to others. And we can consider this as a sort of a functional profile of VOTC in each group, that we can also represent in the format of this similarity matrix, in which we see basically in yellow the categories that are easy to decode, then that they are different, and in blue, categories that have a low decoding accuracy, so that have a similar representation. And then to quantify how similar or different is this functional profile across the groups, I can correlate these matrices. And if I plot this correlation, we see 2 important points here. The first one is that the correlation is significant in the 3 cases, meaning that there is a shared similarity in the functional profile of VOTC across the 3 groups. However, we see that the similarity between the sighted in vision and the early blind in the auditory experiment is significantly higher, suggesting again that the functional profile of VOTC between the auditory and the visual stimuli is more similar between sighted and blind than even between the 2-sighted group. So to summarize what I show today is that the organization, that the categorical organization that we find in the VOTC of sighted people for visual stimuli is at least partially preserved also when the stimuli are presented in the auditory format. And it is even extended in case of early visual deprivation. I showed you that the topographical map of VOTC is shares similarity across the 3 groups, and that the functional profile of this region is also similar across the 3 groups. So unfortunately, in 10 minutes, I couldn't explain all the analysis we did, but we also looked more in detail in our work at which mechanism could explain that, and also at the differences between blind and sighted. So I invite you to go and check our just published paper in eLife. And with this, I want to thank you for your attention, and my supervisor, my group, and eLife for this great opportunity that gave us to share our work. Thank you.
Thanks very much. Sorry, I just had some trouble unmuting myself. Thanks very much. Fab talk. Yeah, brilliant. Again, if anyone has questions, they can post them in this chat down here or in the Google Doc, which is cited in the chat. Brilliant. Okay, so maybe again, I'm just going to start the questions, and it's fascinating data. So, what do you? Somehow we know there's some organization by visual properties as well, right? So we know that from Doris Tsell's work, looking at the basic shape properties across infratemporal cortex in monkeys, and we know because just if you look at convolutional neural network representations, they look similar to the representations in IT or in lateral LOC regions in humans, right? Mm-hmm. So, what given that I guess there are several things. One might be, okay, earlier on in the infratemporal cortex, there's visual information, and then later on you get towards the more semantic or meaning-based information. Or it could be that over evolutionary timescales, it's been driven by visual information, but then it can be, when someone's blind, it can be mapped, it still maintains the same innate representation. Do any of those ideas chime with you, or how do you see it?
Yes, I think that is kind of a mix of the two. So what I believe is that the early visual cortex is for sure a visual region, and then going more anteriorly, we start to find more semantic information, as you say. But still, we see also, like my data are showing that also in the sighted, we already find some kind of information also for non-visual stimuli in this VOTC region, so more anterior region. And what I believe is that in the case of early visual deprivation, so when we have a specific experience of visual deprivation, this ability of VOTC, this innate organization of VOTC just pass through another senses, and it becomes, this auditory representation become even more extended. But yes, I believe that here what we are seeing is a bit a mix between the innate functional organization of VOTC that is definitely there for processing categories, and then also some change that experience apply to this to the nature of this region, making it start to be more processing more non-visual stimuli when vision is not there.
And so here's I wonder if you could try thinking about a different way. So if you looked at the parts of the representation that are different between the blind and the sighted people, do you start seeing anything that looks like an organization by the sound or by the frequency spectrums or of the words that are used to describe it?
Okay, so there are two things I can say there. We checked, like specifically, we test some representational model based more on categories or on low-level properties of the sounds, for example, in the blind. And what we found is that in the blind, we see that the functional profile of VOTC, it's not explained by the pitch of the sounds, for example, but it's more explained by a more high-level functional categorical organization. Then I also looked at something in, I don't know, in here you can see like a clustering, a hierarchical clustering analysis. What I find very interesting is the first line in which I'm basically I'm asking the algorithm to cluster the data in two groups, in each two groups of categories in each of my group. And you see that in the sighted for visual stimuli, that is the up left corner, we have really the animate-inanimate distinction. That is red is basically or the manipulable and big object, and in green we have animal and human. While in the blind, what we find is that the representation, the organization in VOTC is mostly human sounds versus all the rest.
Oh, right, cool. So yeah, Right, so there is some property, yeah.
Yeah, and also this is very interesting because I don't have this data here, but if we look at the temporal regions, so they are really like a region known to process sounds, we find there also incited that the sounds are more like clustered as human versus all the rest. So it seems that this representation in the blind is closer to the one we have in the temporal region.
Yeah, right. So some kind of mixture of a learnt auditory representation imposing on an innate visual representation or something like that.
Yes.
Cool. Very interesting, very cool talk. Thanks very much indeed. So I guess we have to move on now. And so we're moving on to Anantha, who's going to be talking about the role of RhoJ in fibronectin. Oh, hang on, great. I'm going to let Anantha give me her whole title because this is a lot further away from my research area.
Thank you, Tim. I would like to start off by thanking eLife for this forum to discuss my research. So I'm Anantha from Professor Harry Miller's group in University of Bristol, and today I would like to talk about this particular protein RhoJ and its role in the process of formation of new blood vessels, something we call angiogenesis. The process of angiogenesis is actually a very complicated multi-step process wherein endothelial cells, the cells that we see in green here, are supposed to undergo a dramatic change in shape and polarity, whereby they acquire a lot of new properties that help them to respond to the call of tissues to make new blood vessels, which can come out in the form of growth factors. So this change is also accompanied by the ability now to remodel extracellular matrix, which is lacking in the quiescent blood vessels. So this is something that we are really interested in. And we are interested in a group of proteins that enable cells to generally change shape and polarity, which are the RhoGTPase family of proteins. So I would like to go from here to one of our most intriguing observations, which is on this family of proteins of RhoGTPases, of which the most well-studied member is this protein CDC42. It is well expressed in all tissues of our body. However, it has undergone duplication in the vertebrate lineage to give rise to RhoJ, which is much less studied and is mostly only present in the endothelial cell. So it's a lineage-specific protein. As we can imagine, being duplicated, these two proteins share quite a lot of amino acid similarity, and we would expect them to function also in a similar way. However, we found that they are actually working in diametrically opposite directions when it comes to the ability of cells to remodel the extracellular matrix and make new fibrils, something we called fibrillogenesis. So how did we come to this observation? We had initially started off the project to understand RhoJ better, which is a much less studied protein. And we just looked at how RhoJ and CDC42 can be distributed in a primary endothelial cell. And what strikes us immediately is that RhoJ is present in these vesicles, these large vesicles, as well as small ones all over the cell body, which is a distribution quite dramatically different from CDC42. In order to utilize this distribution change to uncover new roles of RhoJ, we took a rather challenging approach to purify these vesicles from primary cells containing RhoJ and see what sort of proteins are enriched in these vesicles. So this is an approach that we call vesicle proteomics, where we can separate out the plasma membrane pool of RhoJ from the vesicular pools by using a density gradient ultracentrifugation, and follow that up with pulling out vesicles containing RhoJ, which was tagged with GFP, using a particle sorter, and then take it through mass spectrometry to understand what sort of proteins are enriched in RhoJ vesicles. So this approach gave us a lot of integrins, which are proteins that bind the cell to the matrix. And one of our topmost hits is this protein alpha-5 beta-1 integrin. And this integrin binds to fibronectin as its ligand and is important for the process of angiogenesis. We were very happy to see that actually the co-localization at the vesicular pool of RhoJ and alpha-5 is very good. In fact, 75% of the vesicles co-localize, suggesting that our approach to purify RhoJ positive vesicles have worked. Now, having these two proteins together in the same vesicle may mean RhoJ can regulate the trafficking of alpha-5 beta-1, or they might be just passively co-trafficking. In order to distinguish this, we actually modulated the levels of RhoJ and saw whether it affects the levels of alpha-5 beta-1 or its distribution in the cell. Unfortunately for us, with multiple approaches, RhoJ did not alter the levels or distribution in multiple different approaches of alpha-5 beta-1. This suggested that maybe RhoJ is just passively co-trafficking with alpha-5 beta-1. However, literature suggests that actually alpha-5 beta-1 comes in two different flavors. You could have this bent integrins which are not bound to ligands, or you could have this stretched out integrins with their tails apart, which are actually bound to ligand. Now, we know from literature that the active integrins are a very small pool of the total, but they are the ones that actually spend a lot of time in vesicles because they have to get rid of their ligand at the low pH of vesicles, and then they can be reused. So basically, we then asked, okay, is there a change in the active integrin alone when we modulate RhoJ? Here, indeed, we could see that when we deplete cells using an siRNA of RhoJ, we can see there is an increase in the level of active alpha-5 beta-1 alone. And we have seen that there is no change in totals. So this really means that there's a conformation-specific change when we alter RhoJ. We have also taken multiple approaches to prove that RhoJ is indeed conformation-specific. And I then had the opportunity to do some cool trafficking assays with Professor Jim Norman at the Beasons, who has pioneered this assay wherein we can actually look at internalization and chase the fate of integrins by biotinylating them at the plasma membrane and watching and taking an approach wherein we can see how they are internalized into the cell at different time points. When we deplete RhoJ, that's the green line here, we can see that the internalization rate alone is actually doubled, and this is true only for the active conformer and not, again, it does not affect the total alpha-5 beta-1. So basically, these experiments prove that the internalization, the point at which the receptor enters the cell, is modulated when RhoJ is not there, and RhoJ seems to be putting a brake to this process. Now, why would this be important for an endothelial cell? We know from literature that this process of trafficking is really important to build fibrils. So it is coupled to the ability of cells to actually direct fibronectin to places where these long fibrils would be weaved. So from this data, our expectation is that when we put a brake on this process, which RhoJ does, there would be much less fibrillogenesis. Indeed, that is really true. When we have depletion of RhoJ, there is the ability of cells to lay out these fibrils is markedly improved, and the converse is true when we express RhoJ in its active state. Now, this was a point where we were again surprised because, as I mentioned, RhoJ arose from the ancestral CDC42 gene, and here we know from literature and as well as our work that CDC42 is a positive regulator of fibrillogenesis. If you deplete CDC42, you get much less fibrillogenesis. This brought us to the question, how do two proteins which are very similar to each other regulate the process of fibrillogenesis in diametrically opposite directions? To understand this, we first thought that RhoJ might have unique binding partners in the endothelium that might help us explain why this is true. So we undertook a study of binding partners in a quantitative way, where we looked at RhoJ and CDC42 binding partners in endothelial cells using TMT mass spectrometry. Again, we were very disappointed to find no unique partners of RhoJ. Actually, RhoJ bound to pretty much everybody that CDC42 bound to. In fact, we could find CDC42 binding partners that were just unique to CDC42, but not RhoJ. It's more recent protein. So the protein we decided to follow up was this protein PAC3, which bound really nicely to RhoJ, but even better to CDC42. To cut a long story short, PAC3 actually behaves exactly like CDC42. It is a promoter of fibrillogenesis. So PAC3 alone enables cells to form fibrils, as also quantified here in a different way. But if you put RhoJ on top of PAC3, it changes direction. In the sense, PAC3 alone drives fibrillogenesis. RhoJ plus PAC3 is an inhibitor of fibrillogenesis, although there is a slight improvement from RhoJ alone. So what all of these data together suggests to us is that PAC3 might be quite limiting in the cell. If PAC3 is available to CDC42, there is more fibrillogenesis. And when PAC3 is taken away from CDC42 by RhoJ, the process is inhibited. So basically, two proteins which are very similar to each other can also drive processes in opposing directions through the same shared effectors if they can compete for the same effectors. Of course, all of this data is actually from in vitro growth of primary endothelial cells. We wanted to know whether this is true in vivo as well. So we collaborated with Professor Akiyoshi Omera in Japan, and they have done this beautiful experiment with RhoJ knockout mice, where we are looking at pictures of retinal angiogenesis, and we can clearly see here that the fibronectin deposition at the angiogenic front here is much more when we do not have RhoJ. This suggests that indeed RhoJ is a negative regulator of fibronectin fibrillogenesis. So what does this all mean to angiogenesis per se? We think that RhoJ activity has to be actively inhibited when the cells have to form these fibrils, when they are sprouting. And this is exactly what one of the growth factors does. And indeed, it has to be reactivated when we have to again stabilize the vessel and form lumen. So this is a brief overview of my work in Professor Harry's group, and it will appear online in a couple of days. So kindly do check out our paper or to know more about this. I would like to stop here and I would like to thank eLife firstly for this opportunity, and I would like to thank my mentors, Professor Harry Miller and Professor Jim Norman, and our funding bodies and collaborators. Thank you so much. I can take questions now.
Hey, thanks very much indeed, Anantha. So I concede that that I'm not brave enough to ask any of my own questions to you, because it is a long way from anything I know about. So I'm going to ask a couple of questions that you've kindly prepared for me. So first, what's the role of other PAC family members, PAC1 and PAC2, in this process?
Thanks, Tim, for asking the question. So basically, PAC3, the PAC3 is a part of a large family of proteins, PAC1 to 6. So basically, we found that PAC1, which is also highly expressed in endothelial cells, also might play a role. It's also competed for, but not to the same extent as PAC3. So PAC1 is also important, but PAC2 does not seem to, even though it's expressed in endothelial cells, PAC2 is not important in this process at all.
Cool. And so Shivani has a question for you. Shivani, can you unmute? And
yeah, I'm unmuted.
Cool.
So is the phenotype of RhoJ knockout similar to CDC42 in mice?
Oh, that's a really nice question. Actually, CDC42 being the ancestral protein, it's way more important in angiogenesis than RhoJ. So if we knock out CDC42, the mice have a very early embryonic death because they cannot make blood vessels right from the beginning. Whereas for RhoJ knockout mice, the effect is much more delayed. So they have a delayed angiogenesis phenotype in the adults. And this difference actually suggests that they have unique functions. So which we have not understood as yet. But indeed, as an ancestral protein, CDC42 has a much higher effect on angiogenesis than RhoJ, and they don't seem to be compensating for each other.
Thank you.
Cool. So another prepped question from Anantha. So can it happen that RhoJ and CDC42 are active simultaneously? And if so, what happens to fibrillogenesis? Actually, before you answer that, we have a new question, but maybe you should answer this instead because it's not from you. So it's from Vinod. Vinod, can you unmute yourself?
Yes, I'm unmuted. I hope you can hear me. So firstly, it's a very nice talk, Anantha, an excellent collaborative initiative. I was wondering if the RhoJ mutant mice are actually viable, because I thought you were showing the retina in one of the slides.
Yes.
And if they are viable, are there other effectors that can compensate during development?
Yeah, so like I was mentioning in the previous answer, basically RhoJ knockout mice do not have any phenotype until they reach adulthood. So there is a slight delay in angiogenesis. So they are completely viable. They are completely fertile. The mice are absolutely fine. We believe that the CDC42 knockout mice are actually embryonically lethal because of angiogenesis defects, and that does not seem to be compensatable by RhoJ. So the delay in angiogenesis that RhoJ causes is, of course, in CDC42 wild type background. So there seems to be unique functions that these two proteins do not compensate for.
Cool. Great. Did you want to go and back and ask about whether RhoJ and CDC42 are active simultaneously, and if so, what happens?
That I should actually say that that's a pretty lame question from my own side, but yeah. So what we know from very recent studies, actually, our collaborators are also doing work on how RhoJ gets activated, and not much is known in the field as such. So basically, what we think is that there is a temporal sequence. So we need RhoJ to be really active in the quiescent blood vessel. And that's important because if you have fibronectin deposition in a quiescent vessel, that's very atherogenic. So you don't want fibronectin fibrillogenesis to happen in a quiescent vessel. And then you want it to happen suddenly when there is angiogenesis. And then you have to stabilize the vessel again when the vessel formation is complete. So we believe that there is a switch between the Rho GTPases at all of these points, and they should not be simultaneously active, basically. So this is what most of the growth factors do. VEGF activates CDC42, inhibits RhoJ, and there are now more reports from our collaborators that there are more stimuli that kind of switch between the two Rho GTPases.
Cool, brilliant. So actually, I think we had one unanswered question for Stefania. Maybe I grabbed all the questions without noticing one. Naomi, is that right?
Yes. This came in the doc afterwards. But all those talks were fantastic. And if we like, we can open it up for all the speakers now. Yeah,
good plan. So Naomi, what was the question for Stefania? And is the participant still online?
Yeah, it's from Vinod. Vinod, you can speak again if you like.
Oh, yes. Do you hear me now? Yeah. So this question for Stefania. So this was on if people can actually identify vocalizations or sounds of animals that they haven't actually encountered before. For example, I was thinking more about kangaroo sounds, which, for people in Australia, maybe that's very familiar, but for someone who's outside Australia, they might not have heard. And do they categorize in similar regions?
You mean if the animals are categorized in similar regions for the sounds in blind and the visual stimulation in sighted?
Yes, exactly. I mean, for me personally, because a kangaroo sounded more like a human was doing the noise, so I thought maybe that's human. So that's why I started thinking about this.
That's a nice question. Actually, I don't know because I didn't have any kangaroo sound in my study, and I cannot respond to your exact question. What I can tell you is that the animal category in general is the one that seems to act more differently in blind compared to the sighted. And my idea, but this is pure speculation, is that actually we use vision to discover, to learn animals, and also like a kangaroo. I never saw a kangaroo in my real life, but I saw on the television, for example, like videos of kangaroos moving, and I can recognize it and categorize it as an animal. What blind people normally do, they either hear the sound or they learn the shape of these animals using small miniatures and then touch. So static miniatures, of course. So either they put them closer to object because they discover them, explore them more as object, or they put them more similar to the environment because, for example, they hear the sound and they probably integrate it with the wind sound or with the other like nature sound. But specifically about the kangaroo that has a sound more similar, maybe also monkeys can have sometimes a sound more similar, I don't know, high pitch, and I don't know, maybe, this is something that it should be investigated, I think, in future.
Thank you.
So I think we're going to wrap up now for the main event. I think there might be some discussion afterwards, but So quickly, so just to say thanks to Uri, Stefania, and Anantha for sharing their work, and to everyone who's still around for listening. We've lost a few participants, but we had more earlier. So our online research talk continues on Thursday, chaired by Dekla Weigel, and we'll hear from Hernan and Carolina and Philip. And so if you want to get updates on this series, if you just follow eLife Community on Twitter, then all the information's there. And yeah, and great. And so we're going to keep this Zoom open for a little longer if there's any further discussion. So people have got more questions, or if they just want to listen to any more general chat between any of the speakers. So that's great. Thanks very much. Okay, Naomi, do you have anything you want to say at this point?
Yeah, I just wanted to say that this is our last week of the scheduled talk so far, but we would like to continue this, not at quite the same frequency, but we'd like to continue to offer slots for people. So anyone who'd like to speak and hasn't had a chance yet, if you go to the web page, which is here, this light green web page, there is a link in the first paragraph to register your interest. And that is what we can then email out to you all to say when we've organized how we'll invite the next speakers, we can say to everyone on that email list. So please do register your interest if you want to speak in upcoming talk slots. And other than that, I mean, I think let's just, Anya has posted lots of things in the chat for you all to follow different news. If you have any questions for the speakers, there's not very many people left online at this point, so I don't want to take people's time if there's no more questions, but perhaps if the speakers have any questions for each other, or if anyone has any questions, we can just move to that now, and otherwise we can end whenever we'd like to end. But I'd also like to say thank you. Thank you, Tim, for chairing, and thank you all the speakers.
Thanks, Naomi. It's fun. It's very, very smoothly organized as always.
Thank you so much, Tim.
Thank you.
Thank you.
Excellent. So do people want to hang around? I can continue asking inane questions if people would like me to, or we can all go about our day's work.
You're welcome to ask if you want to, of course.
So Uri, how do you think, if there's no build up to a voluntary decision, what's happening in the brain that makes the decision then?
No. So I don't think the decision is made in the kidneys, but I mean, I think there is a buildup in the brain that leads to a decision. I mean, if you run some kind of classifier on the brain, you can see that you can predict better and when you're closer to the decision. However, if you look at some of the studies that have suggested that you can tell that what the decision is going to be a second in advance, or maybe even there's some studies that claim maybe even 10 seconds in advance, you can tell with better than chance that there's some information even 10 seconds in advance whether you're going to raise your left or your right hand. If you think about it, it's a little weird. So if the idea is that the brain doesn't know, but that there is clear information in the brain that the decision has been determined 10 seconds in advance. So what's the brain doing for 10 seconds? Just waiting for what exactly, right? So
or maybe I think the people that have said that have maybe suggested something else, which is something about the state of the brain has a probabilistic influence on what the actions are. Right? Isn't it? I mean, and so and those,
I mean, there are different interpretations, and of course, I mean, even those studies that suggested. I'm sorry.
You could tell sort of. I guess you could tell 6 months in advance whether I'm going to choose to eat something hot and wholesome or something light and summery. You know.
Yeah. Yeah. No, I agree. It's not all about prediction, of course. But there is, for instance, some information when we try to really look at predicting, I think a bit more carefully, it might be that you can really, that the prediction ability really rises just close to movement onset and not really well in advance of movement onset, as was thought, or at least as some people have claimed. So that's, I mean, one option is that. The other one is that even if it's not specifically the readiness potential, that is predictive of upcoming movement. It could be that there are other things in the brain, you know, that it's not, I mean, that specific signal is maybe not the right one. You know, it's been known since at least 1965, and it's kind of like people are well versed in it, but it doesn't mean that that's necessarily where we need to look. It could be that there are other things in the brain that are more predictive of upcoming actions than the readiness potential.
So if you look at, like, I don't mean to dominate this, but so if you look at the decision-making literature, and you'll say, okay, maybe one decision is about an action itself, but this decision isn't about the action. The decision is about the good, or the charity, and so maybe it's those regions, that some different region, like the maybe frontal cortex rather than the action system, is making decisions about those in those different frames of reference, and maybe, and so you're just not detecting them with your particular sensor on the EEG. Is that right? Is that what you're saying?
Yeah. So yeah, one thought that we have is it could be some ventromedial area or something like that is involved, and we're actually now trying to do an fMRI version of this similar experiment, an fMRI, to try and really see if there's something that's, as you said, it's just not accessible with EEG, and we could find the information there. Again, I really think we're going to find the information somewhere in the brain. It's not just coming out of the blue, but it seems, at least from our results, it seems to be not the readiness potential that's the important thing here. And I think, again, more than just saying that this is readiness potential or not, I think it's just this idea that arbitrary and deliberate decisions are not the same. And the fact that you can predict, I mean, if you think about it, if you can predict if I'm going to raise my left hand or my right hand, all you need to do is basically, I mean, there needs to be some kind of symmetry broken in the brain. Yeah. So if you're, the fact that you can do some kind of symmetry breaking, that maybe even something like the readiness potential is just a matter of symmetry breaking, you need more than that to predict a deliberate decision, right? Then there's values that go into that, and you know, deliberation back and forth and so on. So we were just trying to highlight, and hopefully we did it in a good manner, to highlight that there are these differences between arbitrary and deliberate decisions. And the fact that you can maybe predict an arbitrary decision doesn't immediately mean that all decisions are like that. And hopefully, if I convinced anybody of anything, maybe I've convinced you of that. So I've done that. I've done my job.
And so, Stefania, how do we? So it seems like as you move away from anterior to LOC or something like that, we're going to need a different way of thinking about the organization of the anterior parts of the infratemporal cortex. How are we going to get that? What are we going to like, as you move towards the hippocampus, we're going to need something that knows an awful lot about what those objects are, if we're going to and how they contribute to our ongoing behavior, if we're going to understand those representations. So it feels like the moment the best we've got for understanding representations is just what they look like. You're showing that there's more than that. Other people show there's more than that as well. But where do we go next?
So I think what would be interesting is to try to put it in a more general view. So, for example, to look at the mechanism explain this, and why the brain does need a region that is more posterior, like VOTC, so that is still involved in visual processing, and why this region would need to process also auditory information, for example. What is the function of this? And yeah, our idea is that, yeah, the brain is all, we know that the brain is all connected, and that there is a sort of intrinsic connection that probably might push these representations. We looked at the connectivity of the brain of VOTC to the rest of the brain, and what we found is that, yeah, there is a sort of like feedback probably information. This we don't know because we didn't check the direction, but that might create some networks for faces, for human voices, all connected together, and making these regions speaking to each other, and then creating sort of brain networks for different categories, for example, such as human or environmental sounds and visual stimuli and so on. So I think that's the way we should look now to understand better how this process is more integrated in the rest of the brain.
Cool. Okay, I'm done with questions now. So if you guys don't have questions for each other, then I think we should wrap up. Thanks very much again.
Thank you.
Thank
Good
you.
meeting. Cheers,
Thanks,
guys.
everyone.